Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Today I am going to give you an introduction to biomolecules. An interesting fact is that our life is composed of lifeless chemical molecules. Those are known as biomolecules. Biomolecules means bio plus molecules means molecules present in biological system you can consider. So biomolecules are chemical compounds found in living organisms. They include organic and inorganic compounds. In order to analyze a living tissue sample and identify a particular compound, first of all, extract the compound from tissue. Then follow different purification methods till the separation of a compound from all other compounds. After that, using various analytical techniques, the molecular formula and the probable structure of the purified compound can be predicted. Let's know different major steps involved in the analysis of organic biomolecules in a tissue as well as analysis of inorganic compounds in a tissue. For the analysis of organic biomolecules in a tissue, you can take any living tissue either from plant source as leaves, or you can take liver from animal source and grind it in trichloroacetic acid using a mortar and pestle to make thick slurry. After that, strain through cheesecloth or cotton. You can get two fractions. One is filtrate and another one is known as the retentate or residue part. Filtrate means it is acid soluble pool with cytoplasmic composition. It contains low molecular weight biomicromolecules with molecular weight less than 800 Dalton. Thousands of organic compounds are found in acid soluble pool, including amino acids, sugars like monosaccharides and few disaccharides, nitrogen base like purine and pyrimidine, nucleosides and nucleotides, etc. Whereas if you consider about the retentate, in that case it is actually acid insoluble pool containing cytoplasmic and organelles composition. It contains high molecular weight, that means molecular weight higher than 800 Dalton, biomacromolecules, including protein, polysaccharides, nucleic acids, that means DNA and RNA. Along with that, lipid is also present in the retentate part, although it is biomicromolecule because it has the molecular weight less than 800 Dalton. You can also use this flow diagram for the analysis of chemical composition in a tissue. Let's know why lipids are found in the acid insoluble fraction or in the retentate part during the analysis of chemical composition of tissues, although they are biomicromolecules with molecular weight less than 800 Dalton. That means lipid is biomicromolecules, it should be present in the filtrate part, but during this analysis process it is present in the retentate part. Let's know what is the reason behind it. Before you know what is the reason behind it, you should know what is the where actually lipid present. If you consider the fundamental building block of membranes are phospholipids, so phospholipids means they are making lipid bilayer in the cell membrane or organelle membranes, and phospholipids actually composed of mainly two things. One is known as the polar head. This is polar head, which is hydrophilic in nature, whereas hydrophobic region is present, which is known as the hydrophobic tails. And tails are nothing, just fatty acid composition. Fatty acid is not soluble in water, therefore tails are hydrophobic in nature. Now let's know what is the actual composition of polar head. Polar head means glycerol phosphate, and this phosphate group is esterified with an organic molecule, either choline, ethanolamine, or ionocetal. Whereas hydrophobic part means, already I told you, fatty acid. It is either saturated fatty acid and as well as it may be unsaturated fatty acid. During the analysis of chemical composition, when tissues are ground in an acid, that means trichloroacetic acid, in a mortar and pestle, the biomembranes, that means cell membrane or organelles membranes of cells, are broken into pieces where lipids as phospholipids are attached and form water insoluble vesicles. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool or retented part, and hence in the macromolecular fraction, although biomicromolecules, it is biomicromolecule with molecular weight less than 800 Dalton. Protein polysaccharide nucleic acids present in acid insoluble pool, they are coming under biomacromolecules and they are polymers. However, although lipid is separated in acid insoluble pool, it is biomicromolecule and it is not a polymer. Now, why lipids are not polymers? Lipids are not polymers because they are not made up of repeating monomer units. Polymers are large molecules composed of repeating structural units or monomers that are covalently bonded together. As for example, if you consider about the protein molecule, this is polymer because it is composed of several amino acids. Now, if you consider about polysaccharides, it is composed of repeating units of monosaccharide. And if you consider about nucleic acids, means DNA and RNA, in that case also nucleic acids are composed of nucleotides, repeating units of nucleotides. Therefore, these are polymers, but lipid is not polymer. It is composed of mainly fatty acid and alcohol like glycerol. These are not repeating units. So we can categorize organic biomolecules into two groups. One is biomicromolecules and another one is biomacromolecules. Biomicromolecules means molecular weight less than 800 Dalton. Macro means greater than, molecular weight is greater than 800 Dalton. Examples of biomicromolecules are amino acids, sugars, nitrogen base including purine pyrimidines, and as well as lipid molecule. So among these biomicromolecules, amino acid, sugar, nitrogen base, these are coming under acid soluble fraction, acid soluble pool, whereas lipid is coming in the acid insoluble pool. Now if you consider about biomacromolecules, protein, polysaccharide and nucleic acids, they are coming under coming inside the acid insoluble pool or in the retented part. This is a list of compounds present in acid-soluble pool and in acid-insoluble pool. If you consider about glucose, glucose is monosaccharide, so uh, it is actually my biomicromolecule, so it is present in the acid soluble pool. Lysine, glycine and other 18 amino acids, total 20 amino acids are mainly present, so they are mostly water soluble in nature. So amino acids are mostly present in the acid soluble pool. These are biomicromolecules. If you consider about fructose, this is monosaccharide, right? So this is also monomer, so my biomicromolecule, and it is present in the acid soluble pool. Sucrose is disaccharide. Disaccharide means in this case sucrose is composed of fructose and glucose and it is water soluble, so it is present in the acid soluble pool. Cane sugar means this is also sucrose, so it is disaccharide. And if you consider NaCl, this is salt. It is also present in the acid soluble pool. Sulfate ion is present in the acid soluble pool. Adenosine, cytidine, guanosine, uridine, thymidine, inosine, and deoxythymidine. These are nucleosides. Nucleoside means nitrogen base attached with ribose sugar or deoxyribose sugar. So this is also biomicromolecule. So nucleosides are present in the acid soluble pool. Ribose is pentose sugar and this is monosaccharide. This is monosaccharide. So obviously it is present in the acid soluble pool. Vitamins, especially if you consider about water soluble vitamins, like vitamin B complex, if you consider vitamin B complex and vitamin C, they are present especially in the acid soluble pool. ATP, if you consider, this is nucleotide triphosphate. So nucleotide is biomicromolecule, monomer of nucleic acid. So it is also present in the acid soluble pool. It's not really a nucleotide. Nucleotide means most of the cases you will find key single phosphate group is present, but in case of ATP, it is in this case 3 phosphate groups present. So it is adenosine triphosphate. Now, if you consider about the starch, cellulose, and inulin, so these are polysaccharides. So polysaccharide means this is polymer. So they are biomacromolecules and present in the acid insoluble pool. Starch is composed of glucose. Cellulose is also composed of glucose but through beta glucosidic linkage. In starch it is alpha glucosidic linkage, but if you consider about the inulin, it is the polymer of fructose, and at the end of the inulin only one glucose molecule is present. So you can consider this is the polymer of fructose. Inulin is the polymer of fructose and it is polysaccharide. Therefore, it is present in the acid insoluble pool. Collagen is protein, so this is also polymer. So it is present in the acid insoluble pool. Enzymes are also protein, mostly protein in nature, mostly protein, except ribozyme. Ribozyme is RNA in nature, so both protein and RNA, they should present in the acid soluble pool. Enzymes are not typically found in the acid soluble fraction because they exist in a colloidal state and are only slightly soluble in acid. So we can consider it is present in the acid insoluble pool. Plasmid means this is DNA, so this is nucleic acid. So nucleic acid is the polymer biomacromolecule. So it is present in the acid insoluble pool. Fat, oil, wax, steroid, phospholipid, they are lipid molecules, but they, although it is biomicromolecules, but it is present in the acid insoluble pool. Similarly, fatty acid is insoluble in water. It is present in acid insoluble pool. Fatty acid like palmitic acid, stearic acid, palmitoleic acid, etc., they should present in the acid insoluble pool. Ribosome, Golgi bodies, these are organelles. So, therefore, they are present in the acid insoluble pool. Insulin is protein in nature, so it is biopolymer, so it should present in the acid insoluble pool. Now let's do the analysis of inorganic compounds in a tissue. First of all, you have to take weight of living tissue, that is known as the weight. Then dry it to evaporate the water and then take its dry weight. After that, fully burn it to oxidize all carbon compounds to gaseous form. That means carbon dioxide and water vapors are released from the burnt substance. This burnt form is known as the ash, which is composed of inorganic biomolecules including inorganic elements like calcium, magnesium, sodium, potassium, etc., along with inorganic compounds like different salts, salts like sulfate, phosphate, sodium chloride, calcium carbonate, etc.
The book, a list is given for the representative inorganic constituents of living tissues, including sodium, potassium, calcium, magnesium, water, and few compounds like NaCl, calcium carbonate, phosphate, and sulfate. Another table is given here as a comparison table in between elements present in non-living and living matter. If you consider here hydrogen, carbon, oxygen, nitrogen, sulfur, these are present maximum in human body as compared to earth's crust. Whereas if you consider about sodium, calcium, magnesium, and silicon, in that case earth crust, maximum amount present on the earth crust. Another thing is that silicon is present in earth crust, whereas it is present in human body negligible amount. Carbon amount is very high in case of human body, which is almost absent in earth crust. Oxygen amount is maximum in human body as well as in earth crust. So in case of human body it is 65.0%, and in case of earth crust it is 46.6%. And another point is that nitrogen is present in earth crust very negligible amount, whereas it is present 3.3% in case of human body. So you can also follow this table.
These are overall biomolecules, means organic biomolecules, inorganic molecules. Organic biomolecules are of two types: micromolecules and macromolecules. Micromolecules including amino acids, sugar, nitrogen base, lipid, fatty acid, glycerol, whereas macromolecules are protein, polysaccharide, nucleic acids, that means DNA and RNA. Whereas in case of inorganic molecules, inorganic elements and inorganic compounds are there. Water is the maximum constituent present in cell. It is about 70 to 90%. Other than that, other organic molecules, if you consider, protein present into 15%, that is the highest amount among all the organic biomolecules. Nucleic acid 5 to 7%, carbohydrates 3%, and lipids only 2%, and ions present in 1%. So in this way, you can also remember the sequence P, N, C, and L. So maximum protein, then nucleic acid, then carbohydrate, and then lipid. Now I'm going to talk about the major complex biomolecules present in the living cells. First of all, carbohydrate or polysaccharides. Polysaccharides are composed of monosaccharides like glucose, ribose, fructose. These are monosaccharides. Primary bond present in between monosaccharides, that is known as the glycosidic linkage. Primary elements are carbon, hydrogen, and oxygen. Examples are starch, glycogen, and cellulose. These are composed of glucose molecules. Major functions, if you consider for the polysaccharides, it is the storage form of energy for short-term demand. It is they are precursors for many organic compounds, structural components of many organisms. If you consider about protein molecule, it is composed of amino acids. Mainly 20 amino acids are present. The amino acids are attached through peptide bonds, CONH bond or peptide bond. Primary elements are carbon, hydrogen, oxygen, nitrogen, and sulfur. Examples are enzymes. Enzymes are mostly made up of proteins except ribozymes, which is made up of RNA molecules. Antibodies are glycoproteins. Receptors are made up of protein. Few hormones are protein hormones are there. Transporters are also made up of proteins. Major functions, if you consider for the protein, this is the fundamental basis of structural and functional basis of life. It is required for the growth and tissue repair. Next major complex biomolecule is nucleic acid. Nucleotides are the monomers of nucleic acid. In case of DNA, it is deoxyribonucleotide. In case of RNA, it is ribonucleotide. Nucleotides are attached through phosphodiester bond. Primary elements are carbon, hydrogen, oxygen, nitrogen, and phosphorus. Examples are DNA and RNA. DNA stores genetic or hereditary information, whereas RNA is required for the protein synthesis. Fourth important complex biomolecule is lipid, which is made up of fatty acid and alcohol like glycerol. In between fatty acid and alcohol, ester bond is present. Primary elements are carbon, hydrogen, and oxygen. Less oxygen, more carbon and hydrogen. Oil, wax, fat, these are uh, examples of lipid. Now, this is the storage form of energy for long-term energy demand. It is membrane component. It is major membrane component, and it can act as insulator. Steroid hormones are made up of lipid. So among these four complex biomolecules, carbohydrate, protein, and nucleic acids are polymers because they are composed of repeating monomers, repeating subunits. Whereas if you consider about the lipid, it is not polymer. It is composed of fatty acid and alcohol, not repeating units. Already I have shown that monosaccharides are linked through glycosiding linkage. Amino acids are linked through peptide bond. Phosphodiester bond is present in between nucleotides, whereas ester bond is present in between fatty acid and glycerol. Let's know in detail about the nature of bond linking monomers. In a polysaccharide, the individual monosaccharides are linked by a glycosidic linkage, and this bond is formed by the dehydration, or you can consider one molecule of water is removed. So one molecule of water is removed so that glycosidic linkage is produced in between two monomers. I have given here maltose as the disaccharide, where two alpha D glucose present in between two alpha D glucose, alpha glycosidic linkage present. Whereas I have given another disaccharide, that is cellobiose, which is composed of two molecules of beta D glucose. Therefore, in between these two glucose molecules, beta glycosidic linkage present. So maltose and cellobiose are disaccharides. So similar way for the polysaccharide, n number of glucose present in case of starch if you consider. So in that case, in starch molecule, alpha glycosidic bond present because it is composed of repeating units of alpha D glucose. Whereas if you consider about the cellulose, it is composed of repeating units of beta D glucose, and therefore beta glycosidic bond present. So you can consider glycosidic linkage two types, alpha and beta, and sometimes you will find in this case you are getting that alpha 1,4 linkage present, but somewhere you will find alpha 1,6 bond also present, especially if you consider about the glycogen and starch molecule. Along with the alpha 1,4 linkage, alpha 1,6 linkage also present. In a protein molecule, amino acids are linked by peptide bond, which is formed when carboxylic group, that means COOH group of one amino acid reacts with amino group of nearby next amino acid with the elimination of water moiety. So this is also dehydration process. In this process, water is removed, H2O is removed, so that there will be formation of peptide bond. Peptide bond means this is CONH bond. Okay, so CONH bond is present in between two amino acids. Phosphodiester bond is formed in between two nucleotides by releasing water molecule. So that means it is a dehydration or condensation process. In a nucleic acid, that means in DNA and RNA, a phosphate moiety links the 3' carbon of the sugar of the one nucleotide to the 5 prime carbon of sugar of the succeeding nucleotide. The bond between phosphate and hydroxyl group of sugar is an ester bond. So you can see here, this is the hydroxyl group and this is phosphate group. So bond between phosphate and hydroxyl group is known as the ester bond. But you can see here two ester bonds present. This is one ester bond and this is another ester bond. So here in the second picture you can see this is one ester bond and this is another ester bond. So therefore, it is known as the phosphodiester bond, and phosphodiester bond is formed in between 3 prime and 5 prime position. So this bond is known as the 3 prime 5 prime phosphodiester bond. In case of lipid, ester bond is formed in between glycerol and fatty acid. In this case also you can see uh, water is released here. This is water. So here this is also water released, and due to that process there will be formation of ester bond. So in glycerol and fatty acid, in between that 3 ester bonds possible, and either these fatty acids are same fatty acids or it may be different fatty acids. So in triglyceride ester bonds are present. If you think this video is helpful for you, kindly like, share and subscribe this channel.